Hello everybody and welcome to my protein synthesis science project. So the first things we're going to be doing is we're going to be talking about comparing and contrasting nucleotides. So first things first, a nucleotide is a nucleic base combined with a deoxyribose sugar or a ribose if it's RNA and a phosphate group. So right here we have DNA. And so this is the main thing that we're going to be talking about a lot besides RNA. So this is like the master plan of the cell. So just for future reference, green is guanosine, yellow is cytosine, blue is adenine, and red is thymine. Also, orange is uracil. Hope I'm pronouncing these right. Here we have the DNA molecule, okay, and then after this we have RNA molecule. So already we can notice some differences. The first one you're definitely going to notice is this one has two strands instead of just one. That's why the DNA molecule is called a double helix. Also, you'll notice RNA doesn't have thymine, it has uracil instead, right here. So to explain in further detail how these things are different, we are going to get down to the molecular structure. So over here I have built two molecules. One of them is deoxyribose, the other one is ribose. Of course, the deoxyribose is on the left because it is missing an oxygen. And for future reference, orange is hydrogen, red is carbon, white is a single bond, black is a double bond, blue is oxygen, and green is nitrogen. DNA has a deoxyribose, while RNA has a ribose, and that's the sugar that's connected to the phosphate group. Okay, so now we're going to get on to nucleic bases. So over here I have built nucleic bases. All 5 of them, and their molecular structure. So over here we have adenine. Adenine is a purine because it has 2 rings. It is also represented as blue in our structure. It is in both DNA and RNA. Over here we have guanine. Guanine is also a purine because it has 2 rings, and it is represented as green in our structures. Over here we have cytosine. Cytosine is a pyrimidinous, whatever that word is. I'll put it on the screen. Cytosine is a pyrimidinous, and it is represented as yellow in our model. Here we have, it's also in both DNA and RNA, same with this one. Over here we have thymine. Thymine is also thymine is also a pyrimidinous. Probably should look up the pronunciation for that. And it is only actually found in DNA, and it's not in RNA. And thymine is represented as red in our structure. Over here we have uracil. Now uracil is one of those particular ones that is only in one as well, just like thymine. It's also a pyrimidinous. And it is represented as orange in our structure, and it's also only found in RNA and not in DNA. Okay everybody, so we're going to be comparing and contrasting the structure and functions of DNA and RNA in protein synthesis. So first things first, we have DNA, and like I said earlier, it is like the master plan. But RNA also has 3 different types, but we'll get to that later. First things first, we're just going to talk about protein synthesis. So what DNA does is it goes through a process called transcription, where it creates a strand of RNA that goes to a ribosome. So what happens is this strand of RNA goes to the ribosome, and the ribosome almost kind of reads it, and it attracts tRNA to come in and deposit these little amino acids, which makes a polypeptide chain, which eventually will make a protein. You've been hearing about RNA a lot, but don't you want to know what types of RNA there are and what their jobs are? Well, that's what I'm here to teach you. So, first things first, like I was saying earlier, during transcription, the DNA makes an RNA called messenger RNA. Now, this all happens in the nucleus. The DNA gets split, and then it gets transcribed onto RNA, which then goes out of the nucleus into the ribosome. So that's our first type of RNA that we have. The second type of RNA we have is, of course, tRNA, which is transfer RNA. So they have nucleic bases, and they're able to read the messenger RNA, and they pair up with it, as this one has. So there's a double blue here, and it's yellow, orange, and blue. And if you look on this side, this has green, and the other side should have orange. So it comes down, reads it, so it makes sure it gets the right amino acid to put on the chain. And we have a third type, though, of RNA. This is rRNA, ribosomal RNA. It is located inside the ribosome. And this is kind of what makes the ribosome work. See, it's just got a whole ton of RNA in it. Messenger RNA comes through the ribosome. tRNA comes in with amino acids based on whatever nucleic bases they have. Nucleotide bases, I think, actually. Whatever the bases they have are. And they deposit it, put the amino acid on the polypeptide chain, and then leave. See, this one does not have one. The ribosome has RNA in it to tell it what to do, and that's basically how translation works. This polypeptide chain will grow up and become a protein. Now, these proteins have to be exact in the amino acid sequence, which is why the RNA and DNA are so important, because they contain the exact information of how to make these, in bases of 3. We might get to that later. Okay, next up, we are going to be explaining the relationship between DNA, proteins, and genes. So, of course, right here, we have a DNA molecule. Now, its relationship to a gene is, part of this is a gene. So, like, for all we know, from here down to here could be a gene. Now, what a gene is it's the specific thing to make a certain protein. It's a specific code. And like I said, codons are bases of three bases. So over here we have a gene, kind of cut out. This codes for a protein. Okay, guys, next up we have the genetic code. Now, earlier I mentioned that pairs of 3 nucleic bases are codons. Now, this is how you decode it and tell what tRNA goes with what codons. So, like I said, tRNA has nucleic bases on them, which I am pronouncing that wrong. I do realize that, as you will notice, that it is correcting me every time. So basically, this is how you decode them. So if you ever get DNA, you can transcribe that to mRNA by looking at their counterparts, except so like A to T and G to C. But instead of T, it would be U. So you transcribe that and you get your mRNA, right? And then you can transcribe that back into tRNA, and you get your codons. Now, to figure out the amino acid chain, you use this thing. Now, with cases of 3, there are 64 different combinations there can be. But there's only 20 amino acids. So, the answer to this problem is some of these codons make the same amino acid. For example, GUU, GUC, GUA, and GUG all make the same amino acid. So that's how that part works. Genes also have a start codon, which is methionine or something like that. Be on the screen. I'll also, I think that is AUG, so it's down here. It is the only one that can make methionine, or whatever it is. There's also stop codons, and there's 3 combinations to make stop codons. And this is what tells the ribosome to stop. Methionine is what tells the ribosome to start. And that's all transcribed in the genes. So yeah, that's the genetic code. So guys, I hope you all enjoyed this little project that I put together, and hope you learned something.